So chapter 20 here all about viruses and prokaryotes. Introduction. Basically, viruses are tiny non-living particles that are made of proteins, nucleic acids, and upon occasion, lipids. Prokaryotes are living unicellular organisms that don't have their genetic information bound in nuclei. Now let's go get more in detail. So archaea and bacteria are both different domains of prokaryotes. Prokaryotes are prokaryotic celled creatures. They are alive. Viruses are not alive. Archaea and bacteria are small, while viruses are super duper small. Archaea are not pathogens, meaning that they don't cause disease and will not kill you. Bacteria can be pathogens, but are not necessarily so. Some bacteria are pathogens and will try to kill you, others will help you. Viruses are always pathogens. They are parasites and cannot reproduce without infecting host cells. Prokaryotes reproduce asexually through the process of binary fission, and they have both DNA and RNA for genetic information. Viruses only have DNA or RNA. Archaea and bacteria are both prokaryotes, but what makes them different? Well, first of all, bacteria have this complex called peptidoglycans in their cell walls. Archaea do not. Archaea usually have different lipids than bacteria do. And archaea have different DNA sequences that are more similar to that of eukaryotes than those of their fellow prokaryotes, bacteria. Archaea live in very harsh environments that have extreme heat or pH levels. Bacteria live in less extreme environments that are relatively comfortable and normal. Bacteria are the larger domain of prokaryotes because they have a greater variety of taxa of organisms because they live in a greater variety of environments. Prokaryotic shapes. Prokaryotes, both archaea and bacteria, can have one of three shapes. If your prokaryote looks like a corkscrew pasta, then it is spirilla. Get it? Spiral spirilla? Yay. So if your prokaryote is shaped like a rod, then it is bacilli. And if your prokaryote is shaped like a ball, then it is cockeye. Prokaryotes have a pretty traditional cell structure. They aren't as complex and don't have as many organisms as eukaryotes, but they are pretty cool. Prokaryotes don't have a nucleus, so their DNA just hangs down the cytoplasm, bunched up as a ball called the nucleus. Prokaryotes are covered by a cell wall for protection, and some have little hairs called pili. These hairs will attach a bacteria to another bacteria, or to a surface. Some also have a flagella, which is a rat tail-like extension that the cell uses to move itself. Viral structure. So all viruses have some form of genetic information. They can have either DNA or RNA, but not both. And every virus has a protein code called the capsid. Some viruses also have additional membranes called the viral envelope. These viral envelopes are covered in glycoproteins. Either the glycoproteins or the capsid proteins will attach to the receptor proteins on the surface of a host cell. By the way, that picture shows a T4 bacteriophage. Bacteriophages are viruses that attack bacteria and only bacteria. So binary fission is the process by which prokaryotes reproduce. Since binary fission is an asexual process, only one parent is involved, not two. That one parent duplicates its DNA and separates the DNA into two as it stretches out. The two sets of duplicate DNA are on each side of the cell. A cell wall forms between the two sides, creating two new daughter cells. Viral reproduction, which is really infection, is a much more complex process than binary fission. There are two cycles in viral infections, the lytic cycle and the lysogenic cycle. All infections begin with the genetic information virus commandeering the host cell. Some viruses, like the T4 bacteriophage, have capsid proteins that will bind to the host cell surface and will inject DNA into the host cell through its shaft. Viruses that have viral envelopes will bind their glycoproteins to the surface receptor proteins of the host cell and will trick the host cell into bringing them in. The genetic information will commandeer the host cell's metabolic system and force it to produce proteins and copies of genetic information. These proteins and genetic information components will reassemble themselves and thus make more copies of viruses. The host cell will be so full of viruses that it will lyse, which is just a fancy scientific name for bursting. as the new viruses escape and go to infect other cells. In a lysogenic cycle, viral DNA is inserted into the host cell system. This into the host cell's DNA. The viral DNA in the host cell DNA is called a prophage. This does not mean that only DNA viruses can have lysogenic cycles. RNA viruses can convert their genetic information into DNA once inside a host cell. This is what HIV does. The prophage will be copied with the host cell's DNA as the host cell undergoes binary fission. The viral DNA will be in all the daughter cells, thus reproducing without harming the host cell. The viral DNA remains dormant in the host cell until an environmental factor will cause the virus to enter a lytic cycle. A virus that can only reproduce by a lytic cycle is called a virulent phage, while a virus that can reproduce by both a lytic cycle and a lysogenic cycle is called a temperate phage. So here's a diagram of both cycles, and you should probably stop the video and study this. So at this point, you're probably wondering why viruses aren't alive. I mean, they evolve, they have organic molecules, and they attack organisms, so they should be alive, right? Wrong. Viruses are not alive because they cannot grow or develop. They cannot independently use energy. They don't independently respond to stimuli. They don't reproduce on their own. They have to infect cells just to make all their components and replicate genes. Nothing else can crystallize, but viruses do. The bad. So what are these things called pathogens? Well, pathogens are those tricky bacteria and viruses that cause disease. Archaea cannot cause disease, and so are not pathogens. The germ theory of disease was thought up by Louis Pasteur. He figured out that bacteria cause many diseases in humans and other animals. You may also remember him as a scientist dude that realized that life cannot spontaneously spring up. Disease mechanisms, meaning how do these nasty pathogens try to kill you? Well, both viruses and bacteria can cause disease by destroying cells. Viruses can also cause disease by disrupting homeostasis, which they do by forcing the host cell to focus on reproducing viruses rather than performing its own life functions. And bacteria, like tuberculosis, will release toxins that also disrupt homeostasis. So how do you stop these pathogens from killing you? A vaccine is an injection of weakened or killed pathogens, and they're usually used to prevent viral diseases, but they cannot cure a viral infection. There are some antiviral medications, but for the most part, they don't do anything, they just sort of slow down the whole dying process. Bacterial infections can be cured by antibiotics because the antibiotics inhibit bacterial enzymes. The good! Yay! So, Malthus said that the human population, or any population really, will grow unchecked unless it is stopped by famine, war, or disease. If population growth continues and is not stopped by these factors, there will not be enough food, water, living space, or any other resources available. All pathogens, especially viruses, control population growth. If you have food to eat, water to drink, a job, and a place to live, then you can thank viruses and bacteria for killing off your competition. Viruses kill the weakest creatures, thus encouraging adaptations that will survive viral infection and increase natural selection. The good that bacteria do. So if you didn't like my sadistic view of the good that viruses do, then you'll be happy to know that bacteria do some all-around good things for us. Bacteria act as decomposers and break down dead organisms and recycle organic materials. Producers are the basis of the food chain and provide energy and oxygen. Nitrogen fixers convert nitrogen gas, which other organisms cannot use, into ammonia, which other organisms can use. Bacteria generally help us humans. Bacteria live in your stomach and help you digest your food. Some bacteria eat petroleum and clean up other toxins. Some bacteria can be used in medicines, and my favorite use for bacteria, they make yogurt. Yum. Well, thank you for watching, and I hope that you learned a lot.